All right, so this video is about protein synthesis, which takes place during G1 and G2 phases of the cell cycle. Before we begin talking about the mechanics of protein synthesis, I just want to review the idea that every cell in your body has the exact same DNA, except for sperm and eggs and adult red blood cells. But all of that DNA is identical, but certainly different cells are different. And the reason different cells are different is because of different proteins being made in different types of cells. So for example, in an emerging red blood cell, you'd have hemoglobin made, and even though it has the genes to make insulin, it doesn't make insulin because it's not the right type of cell. So actually what determines what proteins a cell makes is actually called gene expression. So some genes are expressed in a cell and other genes are not. And the control of which genes are expressed and not is very, very complex, especially in humans, even more so in humans than in simple prokaryotes like bacteria. So let's get down to the specifics. So how does a cell know what proteins to make? The instructions are in their genes. One of the trickiest questions in the next test, the one that's most missed, is what is a gene? So remember that you have chromosomes, or you've heard of the term chromosomes. You have 46 of them, but you make more than 46 proteins. So each chromosome has a lot of information on it, several genes, many genes in a row. So a gene is like an individual piece of information to make one protein. So it's like if you had a recipe book that had hundreds of recipes in it, and let's say it was like a pie recipe book. A gene is just your apple pie recipe or just your pumpkin pie recipe. It is a shorter segment of DNA that codes just for one protein product. So there is a two-step process for protein synthesis. The two steps are transcription and translation. Be really careful in this unit. There's lots of, there's three words that end in "shan," and that just means that there are processes. So there's replication, which is DNA making an exact copy of DNA, and it is completely identical, and that's for the cell to divide. This is different. This is making a temporary, very similar copy of the DNA to make a recipe, or to copy the recipe to make proteins. So we start off this lecture with an analogy. So to transcribe something is to copy it. So for example, if we wrote, "My biology teacher is the best ever," and transcribed it, we might transcribe it into a similar language or like a different font. So do you see here how the first one, this one is in a different font from this one? Or sort of like if the accent was different, if it was a different dialect of the same language. So to translate it would be to change the language completely. Now we chose Spanish because all of your biology teachers at the school you're at took Spanish in high school. So mi profesor de biología es el mejor. Okay, so this is similar to the transfer of information in the cell. The original information is the DNA. The transcribed copy represents RNA, which stands for ribonucleic acid, and the final translated product represents the protein. So we have DNA to RNA to protein. This first process, DNA to RNA, is transcription, and then RNA to protein is translation. So this picture is showing you transcription, and it takes place in the nucleus because, as we learned in the fall, DNA never leaves the nucleus. Now the tricky thing when you're looking at a picture is to figure out, is it showing you a picture of replication or transcription? Because when the DNA opens, it looks a lot like a replication fork, regardless of whether RNA is going to be made or DNA is going to be made. But what you should key in on is if you see uracil anywhere in the new strand being built, instead of thymine, you know RNA is being built, and therefore you know it's transcription, not replication. So look for the bases. So the first thing that happens is that helicase, which is an enzyme you're familiar with, it breaks the weak hydrogen bonds and opens up the DNA molecule. It is the same enzyme that's used for DNA replication, and it acts on the DNA helix. The next enzyme involved is RNA polymerase. Be really careful to specify that it's RNA polymerase, because if you got in the habit during the replication portion of the unit talking about polymerase without saying DNA polymerase, you have to be specific, because DNA polymerase and RNA polymerase are two different enzymes. DNA polymerase obviously builds polymers of DNA. RNA polymerase obviously builds polymers of RNA. And so we have a new RNA strand, which is a primary RNA transcript. This primary RNA transcript is going to be processed and converted into one of three types of RNA. There is mRNA, which stands for messenger RNA, tRNA, which stands for transfer RNA, or rRNA, which stands for ribosomal RNA. In this biology class, we're not going to talk a lot about ribosomal RNA, other than to say that it makes up part of the structure of a ribosome. So there are several things that have to happen to a primary RNA molecule before it becomes really messenger RNA. And so we're going to talk a little bit more about messenger RNA than anything else. So the first thing that happens is that the primary RNA receives a little cap on the end, it's called a methyl G cap, because it's guanine with a methyl group, which is a CH3 attached to it, and it is the start code for ribosomes. It says, hey, start reading right here. I kind of like to think of it a little bit like the little plastic aglets on the ends of shoelaces. Because just like the aglets on your shoelaces protect your shoelaces from kind of degrading and coming unwound, this protects messenger RNA from enzymes that are hanging out in the cytoplasm, whose job is to break down nucleic acids, and they are called nucleases. It helps messenger RNA also attach to ribosome and begin translation. The next thing that happens is a poly-A tail is added. Now, poly just means many, and A is exactly what it sounds like. It's adenine. So a whole bunch of adenines get added onto the end of the opposite end from the methyl G cap. So right there you can see both the poly-A tail and the methyl G cap winding. Okay, then some unused information is removed. And the unused information that's removed, those pieces that are actually kind of cut out or spliced out, are called introns. And what's left behind are called exons. So if you look at this primary RNA transcript, the pink are the introns and the green are the exons. And what happens is that they are spliced out, and the exons are spliced back together. So the final mRNA molecule is actually shorter than the primary RNA transcript because of all the introns that are pulled out. It's sort of like, if you write an essay and your teacher says, oh, you've repeated yourself a bunch of times, you need to edit some things out and, make it shorter. And usually that's like the better essay. Well, in this case, the introns are removed. And when I was learning this, it always bothered me, those two terms. It seemed opposite because it seems like exons should exit. But the way that I like to think of it is that introns are interruptions that don't really code for the protein. So if you think of introns as interruptions, you won't forget which are the parts that leave. The introns leave because they are interruptions. Okay, so the second half of protein synthesis is called translation. And it takes place in the cytoplasm. So the mRNA has to exit the nucleus. And when it does, it finds the ribosome. Okay, now in this picture, the yellow strand right along here, that's messenger RNA. The little cloverleaf shaped molecules are tRNA, or transfer RNA. And on one end, they have 3 nitrogen bases right here. And on the other end, they have amino acids. Sometimes tRNA molecules are carrying amino acids and other times they're not. When they have an amino acid attached, they're said to be charged. So if you hear of a tRNA molecule being charged, that means it is at the moment carrying an amino acid. And remember, there are 20 different amino acids that could be carried on different tRNA molecules. So if you look at these 3 bases on the mRNA, those are referred to as codons. They are complementary to anticodons on a specific tRNA molecule. So each type of tRNA molecule, based on its anticodon, which in this case, if you look at this one, it says UGU, U is complementary to A, G is complementary to C, and U is complementary to A. Now, some of you are thinking, now wait a second, I thought A went with T. Not with RNA. Remember, there is no thymine in RNA, and uracil replaces everywhere where thymine would have been. So now U and A are complementary to each other. Now, any tRNA molecule that has UGU will only carry one type of amino acid. So when the anticodon matches the codon, you end up with the correct sequence of amino acids. And you can see a growing polypeptide chain. In this diagram, the different amino acids are represented by those different colored rectangles. So messenger RNA is attached to a ribosome. tRNA, which is called transfer RNA, matches its anticodon to the correct codon on the strand of mRNA. I think the only thing that's really kind of tricky in this section is remembering which one is the codon and which one is the anticodon. The codon is in mRNA right here, and the anticodon is on the tRNA. And one way to remember that is anticodon even has the word T, or the letter T in it because of the T in the word anti. So that's the way you can remember the tRNA has the anticodon on it. So the sequence of the codon tells the tRNA molecules what order to come in and what order to bring the proper amino acid in. Now, where did the codons come from? They are complementary to the DNA sequence that was in the original DNA molecule that coded for that protein. In other words, that gene. So here is an arrow. This is pointing to GAA. And there on the yellow, the group of 3 bases found on each mRNA is called a codon, and it codes for one specific amino acid. And remember, there's a different tRNA molecule for each of the 20 different amino acids. And again, we are in the cytoplasm. Now, at the very end, or actually at each time when you're adding a new amino acid, there is an enzyme in the ribosome that links amino acids together to form that peptide bond. Remember, we learned in the fall that when the nitrogen or the amino group of one amino acid combines with the carboxyl group of the other amino acids, water is formed, so it's dehydration synthesis, but then we have a peptide bond. And then we have a long chain of amino acids, we have a polypeptide. And once it folds into its 3-dimensional shape, that's when we can say we have a protein. So now what I'm going to show you is how to use a codon chart. Okay, so this is actually a pretty easy thing to do. And once you learn how to do it, you'll really never forget. So one thing students always ask me is, hey, am I going to have to memorize this codon chart? Of course not. Whenever you're given a test or a quiz on this material, we will always provide a codon chart in the body of the test for you. It would be very silly to try and memorize this. So I've thrown up on the screen a little segment of DNA, and I'm going to pause and see if you can come up with the messenger RNA sequence that would be complementary to that DNA. Remember, there's no thymine in messenger RNA. So it would be a GU because A and T, A is complementary to T, G is complementary to C, U is complementary to A, G is complementary to C, G is complementary to C, C is complementary to G. Be really careful because what I see a lot of students do is they copy the exact DNA sequence from above, but just replace all the thymines with uracils, but they forget that they have to make the whole strand complementary to the DNA strand.
I wasn't done.
Lots of students do it.
I wasn't done.
All right, so let's figure out how to figure out the amino acid sequence. So the first thing we do is we look at the first letter. So the first letter is the one on the left. So if we have an A, we know we're in this general segment or section of the codon chart. So then it's AG, so A, and then the second one is up here, so we know we're in this block. And then it is AGU is serine. Now, in this particular codon chart, it gives you the first 3 letters of the amino acid. There is a 3-letter code. These guys actually also have 1-letter codes for when you're getting into molecular biology, but we're going to always use the 3-letter codes. So the first one is serine. See if you can figure out what the next one would be. Okay, so the next one, we go G, G, and then we're now GG, and then GGC is glycine. So it's serine, glycine, alanine, glycine. Now, of course, there's no such thing as a 4-amino acid protein, but this is just to get you using a codon chart. Now, you may notice something interesting. Like if you look at this bottom box, GGU and GGC and GGA and GGG all code for glycine. That's fine so long as GGU only codes for one amino acid and, for example, doesn't code for glycine and arginine. That's okay. This is actually an advantage because if you have a mutation and you have the wrong base, in the DNA, you might still get the same amino acid and it might not affect the protein at all. And we're going to talk in more detail about that later, but that would be called a silent mutation. So this sort of situation builds in some extra insurance against mutations messing up proteins because all genetic diseases are caused by DNA mutations, and some of them can be very deadly, but sometimes you can have a mutation that doesn't change the protein at all. It's kind of like, it's fine if there are 4 different phone numbers that can reach your house, and if your friends wanted to get in touch with you, they could call any of those 4 numbers, but it would be a real problem if the same phone number could call you or your best friend, and then if you're like another member of your friend group was trying to call one of you and ended up calling the other one by accident. So it's okay to have like 4 phone lines in the same house, but not